So we have involves basic sciences. The two important topics that are development of a drug, how to discover a drug, sources, and for clinical trials, phase 1, phase 2, phase 3, how to translate into the real practice. So we have two speakers today. The first talk given by our research lab, an expert in pharmacogenomics. That will be followed by therapy by Dr. who is a senior medical clinician.
Starting the regular presentation, we'll start. These objects. Filter. The discoveries. One, and then if you see all this discoveries, serendipity. However, a lot of brainstorming. So if I can draw the same, the earlier days covered. However, with the evolution of technology, the process of drug discovery became very structured. So before going into the process of drug discovery, good afternoon to everyone. In the next 15 minutes, we'll dive deeper into the fascinating journey of a drug, where a molecule transforms from the bench to the bedside. So across the globe, there is a huge unmet medical need is there. So if you look at, there are limited or no available treatment options that can address the disease condition. So a total of 26,000 diseases were identified across the globe, in which approximately 7,000 orphan and rare diseases are there. And if you look at the total number of approved drugs, it's close to 4,000, and the total number of approved small molecule drugs are closely 2,007. Even the collective number is even make a 1/4 of total number of diseases. So there is a huge gap between burden as well as treatment options. So bridging this gap is a challenging endeavor. But with collaborative efforts, technology can be made to improve healthcare outcomes across the globe. So these are some of the key milestones in the history. So the first rational drug design was discovered in the year of 1909 by Ehrlich and Herta for the treatment of syphilis. So initially, they have used their drug etoxyl, which is personally very toxic to the humankind. So these two scientists changed the structure of the molecule, and they discovered salvarsan that reduced the toxicity towards human beings. And the next milestone is happened in the 1960, where they have identified the biological action along with the chemical structure. So they correlated the chemical structure along with the biological activity. So this is called as the quantitative structure activity relationships. In the mid to late 20th century, we can call it as a biotechnology era, where researchers understand the disease states, biological structures, pathways, transport, pharmacokinetics, and dynamics, all these studies. So here, most of the medicinal chemists use this knowledge to modify chemical structure to influence the drug metabolism, stability, efficacy. So if you look at the process of evolution, in the ancient times, most of the sources are from the natural products like plants and herbs, which are having the biological activities are the only source. Later, the synthesis of organic compounds was started in the era of chemical. Here, the original medicinal chemistry expertise came. They have started synthesizing these molecules. Then, rationalizing drug design process was started. Here, they correlated the chemical structure along with the biological activity. So they have carried out the structure activity relationships and used the computational chemistry based drug. And then biochemical era. Here researchers evaluated biochemical pathways and macromolecular structures, DNA, RNA, protein, even the genetic code, as well as everything was identified. And then the coming era is like artificial intelligence and machine learning. Here mostly we are using deep learning techniques which is used to identify the pattern in the chemical structures, and AlphaFold and ESMFold which are majorly used these days for the prediction of the structure of a protein, mostly for structure based drug design, as well as the generative chemistry artificial intelligence techniques. So these are mostly used for the knowledge based drug design. If you look at the entire process of drug discovery and development, which is a very stringent and a regulated process, each and every step is monitored, developed, and with the approvals, because everything is correlated with the public health concern. So the entire process is divided into two types. One is discovery, and the second one is development. So if you look at the initial steps, target selection, compound screening, and optimization comes under the drug discovery, whereas the preclinical trials and clinical trials comes under the drug development. So these are some of the initial steps. So the first and foremost step is target selection. Here we are using various approaches like genomics, proteomics, metabolomics, various informatic approaches for the identification of a proper target. And then we need to identify the lead. So this can be happen through synthesis, or we can get from the natural sources. And then combinatorial chemistry. So it is a process where we can synthesize a lot of molecules and keep it in a drug bank that can be used for screening purposes. And then biological assay development and high throughput screening. So after discovering the lead molecules, the next process is medicinal chemistry. Once we get the hit molecule, here medicinal chemists will develop so many molecules based on the pharmacophore, and then we'll do the structure activity relationship studies, and finally the synthesis. So once the molecules were synthesized, the next process is the studies. Here mostly we'll check the drug affinity as well as the model as well as the normal, and we can identify the mechanism of action along with the lead candidate refinement. Means in case if any molecule is not up to the marked activity, then we can refine the molecule by modifying the functional group of the structure. And once if we refine the lead molecule, the next we can use animal models. So after a molecule cross all these barriers, the next step is the clinical trial. Here, before going any molecule into the clinical trials, we need additional new drug application and form. And the first phase is mostly this phase not always done for all the molecules, which are usually used to assess the safety of the drug in close to 8 to 10 participants, not necessarily do the process. The first one is the phase 1. Here mostly we used to acquire 40 to 90 healthy individuals, because this trial cannot be done in the disease patients where their immune system is already compromised. So here they acquire the healthy individuals and assess the safety and dosage of the drug molecules. So once the molecule cross this phase 1, the next phase is phase 2. Here we used to acquire 200 to 400 participants, which are like patients. And here we used to assess the efficacy and side effects of the drug molecule. And the studies are usually carried up to 2 years. And the next phase is phase 3. Here a large number of population, like 1500 to 2,500 participants will acquire. And here we used to compare the efficacy of the drug molecule with the treatment and also check the adverse drug reaction. These studies can be done from 1 to 4 years. So once the molecule crosses all these three phases, we need to submit the new drug application form. Here the entire data will submit to the regulatory agencies. The FDA will review all the information. In case if the molecule crosses all the barriers without any toxicity side effects, and if the molecule is very, then they will give the approval. Once the drug was approved from the FDA, the next is post-marketing surveillance, like phase 4. Here the studies are long-term effects study of the drug molecule in a huge number of populations. So, but if you look at the journey of a drug molecule, it is like very stringent process and highly resource. The average time to require to develop a drug molecule like 10 to 15 years and $2.5 billion investment. So the image it seems like valley of death, because the attrition rate is very high. In case if you start your process with 10,000 molecule, it is very difficult to reach one to the market. Here, if you look at, if you start the process of the preclinical trials with the 10,000 molecules, the approval may be 1 or 2 molecules. But here the advantage is we are having a huge chemical space. Almost 10 to the power of 60 possible compounds are exist in the chemical space. Yes, if you want to do any kind of drug repurposing, we can use those drug data banks. Basically, why this drug discovery is important. So far, we have identified so many targets, but collectively not equal to 25% human proteome. Still 85% of the human proteome is currently undruggable. So in that some KRAS targets are there, some targets doesn't have any ligands or drugs. So there is a huge scope. Still we can improve this drug discovery by employing some computational tools to explore this 85% of human protein. So what is the role of informatics in, if you look at in any step, informatics plays a major role like target identification. They are mostly used to analyze the genomics data, proteomics data, as well as the integration of pathways. So in every step, the bioinformatics will be useful. And if you look at the chemi informatics, it will be useful for the lead finding and lead optimization. Here we can study the quantity of the molecule, and we can predict the in silico admit properties, bioisosteric designs, screening, descriptor calculation. In every each and every step, informatics will be helpful. So there are different approaches are used in drug discovery. The first and foremost thing is serendipity, chemical modification. So mostly serendipity means everyone will give the same answer like luck, but I don't think so. Luck is not the serendipitous. Sometimes chance favors the prepared mind. So the first serendipitous discovery is like penicillin. We all know the story of how Fleming discovered the penicillin. So usually he used to work with Staphylococcus. So one of the mold contaminated with the plate, and the metabolite which is secreted from that mold showing the inhibitory activity. That he discovered the compound. The second approach is chemical modification, so which is a traditional method. Here mostly we'll start with the active compound which is synthesized with the modification that called biology. So this is a kind of safe process because if any molecule is showing good activity, if we refine little modification, that molecule biological activity. So in case if you look at this molecule tagamet and azotate, both are like almost structurally similar, but this Zontac was slightly modified from the tagamet. But what are the limitations with chemical modifications are end up with something similar. So if you look at the first molecule and the second, almost similarly, a minor differences are there in the functional groups. And the next approach is screening. Here we are using so many filters to cut off the molecules like cleanness rules and toxicity coefficient, pains and in silico admit properties, everything. Or sometimes we can do the virtual screening with the predicted target along with the ligand data. And the traditional drug design here starts with a validated biological target and ends up with a drug that optimally interacts with the target and triggers the desired biology. So here, if you look at the problem statement, mostly histamine triggers the release of stomach acid. So if you want to design any inhibitors that suppress the stomach acid, you can go with the antagonist which is similar to histamine. So here they have synthesized some of the analogs which are closely related to the histamine molecule. So obviously they can suppress the release of stomach acid. Before doing like any drug discovery process, drug likeliness to follow. So that Lipinski rule of 5. For example, if you are selecting any molecule, the molecule than 500, the partition coefficient is less than 5, and the number of hydrogen bond donors is less than 5, and the hydrogen bond donors are less than 10. So we should follow these rules to design a drug molecule. So apart from these Lipinski rule of 5, there are several other rules like MDDR, Weber, Goose, Red barrier, and CMC 50. So each rule is having their own conditions. If any molecule is showing more than 2 or 3 violations, we can skip the molecule. So why after following this stringent process, still many of the drug discovery projects are going to fail? Because 44% of preclinical projects are failed due to optimize the line. So mostly in the very beginning, we used to identify the hit. From hit to we optimize the lead. Here majority of the projects are is 22% of preclinical failed due to the poor initial target. And remaining 34 are just like regulatory affairs. And so considering all this, the initial two steps, target selection, discovery are the key most steps in the drug discovery. So what are the criteria a proper target as well as lead optimization. So identifying the biological origin of a disease and the constant story of a medicine. And here, note, not every target is equally capable of affecting the course of it. So we need to identify such a target. For example, if you want to target a glucose metabolism, then any glucokinase, that doesn't work well. So the glucokinase by the isokinase. So the process cannot be hard. In such case, we need to we do not select those kind of targets. We have to skip. So always we need to select a proper target. So it has to be very specific, a molecule or a biological. And there are some key properties to select the ideal drug target. The first thing is the disease modifying and has proven function in the pathophysiology of a disease or not. And modulation of the target is very less important. If the druggability is not obvious, or a 3D structure or three-dimensional structure for the target protein or a close homologue should be available for it. For example, KRAS is a better example. It doesn't have any active site. So whatever so far identified inhibitors like cetuximab or adagrasib are the covalent inhibitor. And target has a favorable assayability. In case if there is no assay to detect the activity of a drug molecule, useless. So target expression is not uniformly distributed throughout the body. For example, if target is present in the normal cell as well as, then it is very difficult to target such kind of targets. And moreover, a target or a disease-specific biomarker exists to monitor therapeutic efficacy. Apart from this, there are several biochemical targets. So among all the drug targets, G protein coupled stands in the 45% of drug targets, followed by the enzymes, hormone factors. So if you want to hit the apple or head, it totally depends on the skill set that you acquired. So always selectivity is important. For example, if you want to design any antibiotic, that has to selectively target the bacterial enzyme, not the human or mammalian enzyme. So always we need to maintain the selectivity. And finding the lead. So in the very beginning, plants and microbes and marine world or nature itself is a vast repertoire for the drugs. Screening those natural products for the identification of a lead molecule. But never use a crude extracts. Always use, if you identify any crude or any plant source is showing some good activity, do the purification to determine the structure of that molecule. Once if you identify the structure, you can use the same kind of ligand for the synthesis. So it's a kind of safe game. Always use structural similarity to a natural ligand. If you look at vincristine, vinblastine, doxorubicin, all these are comes under the natural ligand. If you keep those molecules as a constant and if you change some functional groups, so obviously you will get some kind of better activity. And day by day, even the combinatorial chemistry is also improving. People are synthesizing so many molecules, the drug banks, which are useful for the repurposing strategies and screening purposes. And screening the synthetic bags. So most of the pharmaceutical companies have prepared and they're keeping in the database. So these are stored in the freezer or cataloged and screened. In case if you identify any new drug, if you want to screen those molecules, they can provide. And once if you identify any molecule, the next step is we need to study the structure activity relationship. For any molecule, the entire molecule does not contribute the activity. A precise portion can contribute to the activity in the biological system. The precise portion is usually called as a pharmacophore. So we need to identify the pharmacophore in the molecule. If you look at the computer-aided drug design, so mostly the small molecules are designed in the two. One is structure-based drug design, and the second one is ligand-based drug design. So mostly, if you know the protein structure, or if a three-dimensional protein is available and you are having the ligands, so this process can be used for the structure-based drug design. And we are having the three-dimensional structure of protein as well as there are no ligands, then the process comes to the de novo design. And if we do not have any known protein structure, but we are having some kind of ligands, so that process comes under the ligand-based drug. If there is no known protein structure and there is no known ligands, then no use of computer-aided drug design. So if you look at the flow chart of structure, in the initially we used to take the ligand database as well as dimensional structure protein, which were from the protein data bank. Here we need to screen all those molecules against the accident, and then we have to perform some kind of dynamics based on the pose and the binding energy, identify some of the hit molecules. So here some of the couple of examples which are discovered through structure-based drug. All this comes under from the structure. And the next process is ligand-based drug design. So if you take nanocar or bench, BMW, anything, the common features make functional car is like engine, wheels. These are the common features. In a chemical molecule, we can call this common feature as a pharmacophore. So take that pharmacophore or identify the pharmacophore and map all the chemical molecules against that pharmacophore. Identify some of the. So here, start with the database and some of the known molecules which are showing good activity. If you map this database with the known activity compounds, you will get molecules. And these are a couple of examples which are identified through the ligand design. So mostly like being a biologist and a medicinal clinicians used to have some kind of preconception that drug discovery is a process belongs to a chemistry person, but it's like a preconception. Here we can see this our what a clinician do, what a biological researchers can do, what a medicine can. So as I mentioned earlier, the initial two steps, target selection and field optimization. These are the key steps. In these two, target selection is the most important where a clinician biological can identify. But the role of medicinal chemistry is only optimization, activity relationships, and in case optimize the molecule only that can. So here, if we want to see the medicinal chemistry, the alternative approach is drug repurposing. Here, not much knowledge or not required on chemistry. So drug repurposing, sometimes they call it as drug repurposing. So here, any drug is having some kind of polypharmacology. Means a molecule can show one or two acting more than one activity. Are usually called as polypharmacology. Of the drugs are having same kind of phenomena. So here we used to take phase 2 or beyond the phase 3 molecules for the drug because the in safe side. All the established pharmacokinetic parameters, tolerability and safety parameters, everything was there with the phase 2 molecule. And moreover, we can reduce the timeline. If you look at the de novo drug discovery, the required time is 10 to 15 years. But with the drug repurposing, we can reduce at least 7 to 8 years timeline so that we can discover more number of drugs. So these are some of the repurposed. Uh, I think I think everyone is aware of the thalidomide. So mostly in the very beginning, it is used to give the pregnant women. But after a certain period of time, it causing some birth defects. So the drug was banned. And later, the same molecule was repurposed. Now it is using for some treating some kind of cancers. And one more blockbuster drug is sildenafil, blue pill. Uh, Generally, it's discovered for the treatment of cardiovascular disorders, but the preclinical and the clinical trials, all the results showed a better treatment for the erectile dysfunction. So now it is using for that treatment. And apart from these two drugs, there are several other drugs for repurposed against different indication. So for doing this drug repurposing, a little knowledge on informatics and is enough to do this drug repurposing. So drug repurposing is the process where we can reduce the time, cost, and resources. So we are having a huge number of drug banks in the chemical space and try to explore them. Obviously, a very interesting set of compounds that were waiting for the right disease. So these are a couple of patents of mine from the US, Europe, and the World Intellectual Property Organization. Thank you very much.
So, very good afternoon. It's very nice to be back here. Such an lucid illustration of the journey of a drug from its the idea of inception to its clinical application. So, I will confine myself for the next 20-15 minutes about the role of targeted therapy in cancers and how we have evolved from moving away and away from chemotherapy towards the targeted therapies in cancer, and that is how we have reached an era of precision oncology in cancer. So I will take you through the genetic basis of cancer. Like that is probably considered as one of the major discoveries of oncogenesis, and how precision oncology and has moved in, and how targeted therapies have now become the way to go. I'll take one example, that is lung cancer, because it is considered as one of the most diversified molecular cancers, molecular cancer with molecular landscape. Among the lung cancers, I will take EGFR, which is considered as the sole driver in lung cancer. Lastly, we'll move to the ideal targets and ideal drugs. I think a part of it, or rather, most of it has been already covered in Dr. Ponchandra's talk. And then we have our own bottlenecks and why we are not able to, stride the mountain as good as the West. So that is how our way ahead in Indian scenario is. So the biggest disadvantage of conventional chemotherapy is its inability, put it in very terms, is its inability to distinguish cancer cells and normal. That is the reason why we have most of the side effects that we see from chemotherapy. Name it, whether it is myelosuppression, immunosuppression, mucositis, vomiting. So the predominant side effects of chemotherapy occur because of the fact that it's unable to distinguish cancer cells. So the normal cells bear the aftermath or bear the side effects. So slowly and slowly we are moving away from the broad spectrum cytotoxic to target drugs. So you can actually name very few diseases on your finger where we have conventional chemotherapy still playing the major role. Now, when we talk about the targeted drugs, two important features, two important advantages that we have: the high potency drugs and of course the low toxicity. When I say low toxicity, I compare it with because the targeted, we will discuss that later. So the first approved targeted therapy, one of the blockbuster molecules, turned the pace of oncology, imatinib. All of us know how imatinib has ushered in the era of targeted therapies, and the first drug that is approved, first targeted therapy that is approved for chronic myeloid leukemia. And now, patients with chronic myeloid leukemia, they have this on par with their peers who do not have CML, age adjusted. Their life expectancy equal to the age adjusted population, even though they now we have moved to an era where we consider something called treatment free remission. So in patients with chronic myeloid leukemia, if this for a particular time, we tend to take them off therapy. That means with CML diagnosed, that is, or does that achieve set criteria of molecular remission, then after 5 years, stop the treatment. So this is very important because when we were students, treatment for CML was, and a female patient, productive age, was not allowed to continue because they are on imatinib. So just imagine that these patients, especially female patients, or say young patients, they will have a fully functional, productive family with the disease under control. That is the reason why these targeted totally changed the course of treatment. Now, I think I will very briefly classify these targeted drugs or molecules as small molecules and macromolecules based just upon their molecular weight. But if you look at the properties, the small molecules and macromolecules properties. One important thing, everything is by virtue of their. For example, if you take the off-target effects, the off-target effects are more common for these small molecules because these will target the other targets which are present at. But that is very rare with the macromolecule. If I have to give you the examples of the most of these signal transduction inhibitors, like all the tyrosine kinase inhibitors, are considered small molecule, whereas the proteins, the antibodies, the vaccines, these are all the macromolecules, molecular. So the off-target effects are very much minimal with these macromolecules. Are more, and the penetration into the cell. And one important feature for us as oncologists is sequestration of the blood-brain barrier. So the drug penetration into the brain is a very important, it's perhaps a problematic one, and we have not reached such success with respect to the penetration of these drugs, chemotherapy, or targeted drugs into the CNS. So brain has always eluded us. To ask if the drugs that we use for managing the glioblastoma, the central nervous system, are hardly few. It's just because the central nervous system is tested away from these drugs or the targeted drugs. So the penetration into the brain is also one. And as you can see, these small molecules have good penetration. For example, I'll take CML itself. So one of the first generation tyrosine kinase or small molecules is the imatinib. Later on, we had the second generation drugs named dasatinib. If you look at these, the drug dasatinib has got almost 300 times more penetration nervous system compared to imatinib. So this evolution of these targeted drugs is also one of the important features for the success of CML. And the main difficulty, if you look at in the development of these, that macromolecules are technically difficult to develop because we have to take so many things in. The development of these small molecules is relatively easy, but they take a longer period of time because you have to also study their off-target effect whenever you are dealing with a small molecule. Look at this timeline, the timeline of development over the past 2 decades. A lot of molecules, and if you see, some of them are very specific, but some of them actually inhibit 2 or 3 other molecules. So this is also very important because let us say if we have a molecule inhibiting a specific pathway, and in the specific pathway, let us say it inhibits at 2 different levels. I think with this thing. So all of us know this, right? All of us have been learned this during our biochemistry, biotechnology, molecular pathologies, right? These are the various pathways which are active in the cell. And for an oncologist, whenever we have a drug which is inhibiting a particular pathway, or at a particular point of the pathway, the cells, the cancer cells develop mutations in the downstream pathway. So they escape that inhibition, and then that is how they develop resistance. So for us, if you have multiple kinase inhibitors or multiple targets, or if a drug is able to inhibit sequence of these targets or a series of these targets, then probably the inhibition would be more fruitful, and probably the effect of the drug on the cancer cells would be more fruitful. So in this one cartoon, I tried to put together 2-3 pathways. One is the predominant, like what Dr. Punch has elaborated, is the A pathway, and there is another RAF MEK pathway, and there is JAK-STAT pathway. And in the cell cycle, whenever the cell transits from the G phase to F phase and then to the M phase, we have enzymes called cyclin-dependent kinases. We have targeted these cyclin-dependent kinase inhibitors are also now available. So we can target the cell cycle, at the same time we can target either of these pathways. So if you look at those tyrosine kinase receptors which are present on the topmost end of the cartoon, we have various receptors. All of these are predominantly tyrosine kinases. They have an extracellular domain, they have an intracellular domain, and they have a transmembrane domain. The intracellular domain is nothing but a phosphorylation domain. So these receptor kinases which have a tyrosine residue, they just phosphorylate the downstream pathway. Once the downstream pathway gets activated, then the cancer or there is oncogene addiction. I'll just come to that concept a little now when I take you through the lung cancer example. So this is regarding the hallmarks of cancer, as we always go back to the multi-targeting or multi-pronged approach to treat cancer. These are the hallmarks of cancer. So where you have the ability of the cell to get replicative immortality, or the ability of metastasis, angiogenesis, invasion, because one of the hallmarks of cancer is to evade cell death, at the same time grow and invade other organs. That is the metastasis. And unfortunately, the primary as well as the metastasis, not only they differ from each other, the metastasis at various sites also differs from each other. The metastasis today differs from the metastasis tomorrow. So this is how cancer poses difficulties in order to overcome and treat. And similarly, if you look at the mechanisms of drug resistance, they also develop drug resistance in each of these pathways. So each of these hallmarks are also, so I mean, what we try to do is that we try to put the hallmarks of cancer, and in each of these we have drug developments. At the same time, those pathways which alter in order to become resistance. That is how the tumor evolves, and of course, our understanding of this also keeps evolving. Now, I took lung cancer as one of the examples to depict the evolution of targeted therapies because of its diversity. I could have taken acute myeloid leukemia, acute promyelocytic leukemia, non-Hodgkin lymphomas, cholangiocarcinomas, you name it. Then there are targeted therapies which are available. But this is one example because this has actually showed us, lung cancer has actually showed us that there is something more and more beyond what we actually look at. And over the past 10 years, scenario of lung cancer has extremely changed. I just show. So for a stage 4 lung cancer, these are the current treatment options. Of course, we offer smoking cessation, systemic therapy. The options include chemotherapy plus minus anti-angiogenic therapy. Again, anti-angiogenic therapy is also a type of targeted therapy. We offer targeted therapies or immunotherapy alone or in combination with chemotherapy. So now for a particular patient, what would be the ideal strategy is something that we choose among these three. Now, if you look at the genomics or the targets that we have in lung cancer, the basic histological differentiation as adenocarcinoma, squamous cell carcinoma, and small cell carcinoma, gold standard even today. But what is important is a subset of these adenocarcinomas, they are genetically simple. When I say simple, it means that there is this oncogene addiction or a driver addiction of the cancer. So the cancer cells are addicted to that particular driver. This is quite commonly seen in around 55 to 70% of East Asian population. Very, very relevant our population or our Indian scenario. Now, and unlike the West, where you only see around 15 to 20% of the cases, and their mutational burden is not so high, and they are not related to environmental carcinogenesis, tobacco carcinogenesis. These adenocarcinomas, which are oncogene addicted, are predominantly seen in non-smokers, female patients. Asian cohort. So if you look at that, that fits into our population, Indian scenario. So again, if you look at the targets which are available in adenocarcinoma versus squamous cell carcinoma, if you look on the left-hand side of the cartoon, you can see that there are no actionable mutations in close to 90% of the squamous cell carcinoma. When compared with the adenocarcinoma, in the adenocarcinoma, you have around only 20%, around 30% of the adenocarcinomas do not have an actionable mutation. The rest of the 70% will harbor some sort of actionable mutations. So identifying this is very, very important. If you look at Asians versus non-Asians, so on the left-hand side, you can see the concentric circles. The outer circle is the Asian population. You see that around 40% of these Asian population harbor that EGFR mutation. And the rest of the 20%, they don't have any actionable mutations. The rest of the 20% have some sort of actionable mutations. Go back to the inner circle. In the inner circle, close to 45 to 50% of the patients do not harbor any actionable mutations. So being Asians, East Asians, and non-smokers, that should actually trigger our minds whenever we are actually dealing with these patients, that there might be a possibility that these patients will be harboring some of these actionable mutations. Now, this is a changing landscape of therapy. Concentrate on the lower half of the slide, where you can see patients who are having driver mutations, and this is the approvals. So way back in 2009, we had targeted therapies approved for EGFR, and now we have so many other targets which are available in first line as well as in the second line. So we have come a long way, and this is only for lung cancer. So identifying the driver or that oncogene which the cancer is addicted to is very important. So this is a multi-gene panel NGS. So whenever we see a patient with a metastatic lung cancer in our OPDs, this is the bare minimum. So we send these, or we actively check for these alterations, molecular alterations, along with an immune biomarker. So this is the bare minimum, and we do this in our hospital regularly. So all patients, metastatic lung cancer, this is a must in the current era, and we use various methods. You can use the PCR individually, or you can use a multi-gene panel NGS. So in our hospital, we use a multi-gene panel NGS for detecting these molecular alterations. And this is something that we have developed ourselves. So when we put together everything, we have a patient with metastatic lung cancer, a diagnostic workup is complete, then we molecular profile the cancer. Once we do the molecular profiling, we again sit with that molecular and we try to identify which is the driver. I'll show you how we do that. Once we identified which is the driver, then look at this. Look at the series of molecules which are available. Not that they are, not that all of them have equivalent efficacy. There are ifs and buts in all that, but we sit together, we look at the molecular profiling report, and then try to take a decision on which particular targeted therapy are we going to subject this patient. And again, as I told you, therapy switching or combination or rebiopsy of these will be done at various stages of the tumor progression. So during this journey of the patient, each and every time point, we keep doing this molecular profiling. So I just want, I actually put 2 to 3 studies together. A cross-trial comparison is not advisable, but I just wanted to show you the fact that when you have an oncogene addicted cancer or a driver addicted cancer, you can look at the survival curves. When we were residents, we hardly had any of these, in our mind. So if you look at EGFR addicted cancer, median survival of the patients crosses around 40 months. When you have ALK or ROS fused or addicted cancers, their median survival crosses to around 60 months. So these are fourth stage metastatic lung cancer patients who are harboring this oncogene. And the reason why we keep looking for this is because, as I told you, Asian population, the incidence is quite that. Among this lung cancer, I will just take you to one driver, that is EGFR, that is epidermal growth factor. You can see that the gene which is present, the EGF, the structure of the EGFR on the right hand side of the cartoon. And I will just take you through the journey as to how it paved way for understanding lung cancer better. How, keeping this in mind, we have understood the concept of oncogene addiction or driver addiction and how we have developed molecules. Now, whenever we have an oncogene addiction, we have a suitable molecule available for it. If you look at the European Society of Medical Oncology, for first-line treatment of EGFR mutated NSCLC, it is no longer chemotherapy, targeted. This first line, either you use geftinib or erlotinib or osimertinib. These are all small molecule inhibitors. So the first line has changed from chemotherapy once upon a time and for the past 2 decades. So whenever we have whenever we identify an EGFR mutation or not, that is non-small cell lung cancer, first line choice is not chemotherapy, it's targeted therapy. Now, which one to choose among these? Of course, depends on the patient and a lot of other factors there. We'll discuss about that as well. So if you see, if you see the mutational landscape or molecular landscape of a metastatic lung cancer, the traditional view was only 3 histologies: endo, squamous, small cell. Later on, we identified that there was KRAS, which is one of the mutations. And in 2004, that was the time period when EGFR came in. 2003, 2004, people detected that EGFR could be an important driver or could be an important target. And in 2009, EGFR inhibitors were first approved for the first line setting. So this is how, and look at 2021, this is a data from Sloan Kettering, and these people have identified that these are all the mutations which are available, which can be identified, and majority of them have targets. Majority of them have drug molecules which are available now to target. So we have moved quite a long way, and this is the EGFR signal. It's just the tyrosine receptor kinase which simultaneously activates RAS as well as PI3K pathway. And at the end, if you can see, it also confers a proliferative advantage to the cell and a survival advantage to the cell. Usually, these are mutually exclusive. And the basic concepts of pathology dictate us that when you have a proliferative advantage, you might not have the survival advantage. But this is slightly different. So both class 1 and class 2 advantages are available, are conferred upon the cell by activation of EGFR. How does the cell get addicted to it? Because of the various activating mutations. So you can see this is the EGFR gene, and The most common mutations that occur are exon 19 deletions, which constitute to around 45% of the total EGFR mutations, and a point mutation called L858R, which occurs in exon 21. This accounts for another 40% of the patients. Whenever these mutations occur, the EGFR receptor is constitutively activated despite a ligand. Normally, whenever the ligand gets attached, only then the activation occurs and the downstream pathway gets activated. But when you have a mutated EGFR, the pathway gets constitutively activated. This is how the particular cell gets addicted to that mutation. So this is the concept of oncogene addiction. And so these are the again, these are the various exons which are commonly involved. And as I told you, exon 19 and exon 21 are most commonly. If you ask me which is better, perhaps exon 19 they do well. This is very important for us also because the drugs that we use, the small molecules that we use, have differential sensitivity to those exons. So identifying that is also very important. So whenever we look at an NGS report, I'll show you. We also look at what is the exon, what is the allelic burden of the mutation, and which drug to use. Because the sensitivity of the drug and perhaps the development of drug resistance is also dependent on the exons that are mutated. And how common is EGFR? Yes. I told you, if you look at the diagram on the right hand side, never smokers have almost close to 50% of the never smokers have EGFR. Among the various EGFR mutations, majority of them are exon 19 and exon 21. There are other less common mutations. Once in a while, we keep seeing these also. These are the EGFR TKIs. So learning everything about EGFR is important because we have drugs which target the EGFR mutation. So these are the first generation drugs which are geftinib and erlotinib. Second generation drugs are afatinib and dacotinib. And third generation, the current generation drugs are osimertinib. So people consider osimertinib as one of the most potent and one of the most efficacious TKI because of its covalent binding. It has an irreversible binding, it is more potent, and it has got a very high propensity for CNS penetration. So none of these TKIs, erlotinib or geftinib, science, when we were students, people with metastatic lung cancer, brain metastasis, always excluded science. We didn't know how these patients actually behave with this till osimertinib. So it is said that osimertinib has a 60% intracranial disease. So the studies which I have showed you, where the median overall survival of the patient goes to 40 months, is including brain metastasis. So that is how osimertinib has actually, turned heads of all the because of its very good CNS penetration ability. Now, again, another timeline where how the TKIs have evolved, and as you can see, the incidence of the EGFR mutations, Asia has a good chunk of patients. East Asia has a good chunk of patients, close to 40% of all the patients actually harbor EGFR mutation. And yes, people have compared EGFR TKIs with chemotherapy and have proven the superiority in various lines, be it second line or in the first line. The list of the trials with various EGFR inhibitors and chemotherapy, superiority was. Among the EGFR TKIs also, the third generation TKI, that is osimertinib, was shown superior to the first and second. Now, all is not so well. As I told you, when you target a target or you inhibit a particular step of a pathway, the cancer cell or the tumor develops resistance by mutations in the subsequent pathways, or there will be a development of another driver, or there will be a clonal evolution of another driver. Or usually, whenever we have signs of resistance or whenever there is clinical progression, we always try to go back and see what is the driver. Has the driver changed, or has the tumor acquired a new molecule, a new mutation, which has now emerged as a major clone? So I will, so understanding this clonal evolution, you can see when you have an EGFR dominant clone, you have given an EGFR, as you can see, there is an evolution of another clone which is resistant to that EGFR TKI. So you have two divergent clones, one is a small clone and one is a large clone. Now, again, when we do the biopsy and assess what is that particular clone harboring, we probably might target. This is how we try to understand the mechanisms of drug resistance whenever a patient is on this molecule. Now, there's been a take. I actually okay. This is one of a patient with gallbladder cancer, poorly differentiated cancer, and it was metastatic to begin with, liver mets, nodal mets, and infiltrating the D1, D2. Patient presented with obstructive jaundice, and we actually sent at the baseline we actually sent multi-gene panel NGS test to identify what he was harboring, because it was a poorly differentiated carcinoma. Normally, we have adenocarcinomas in the gallbladder. Very rarely do we get poorly differentiated a mutational assay. Here, what we found was this patient was harboring FGFR1 amplification, a PI3K mutation, TP53 mutation, and he was not having a significant mutational burden. It is just 5 mutations per megabase. So, at this point of time, we do not have any robust, clinically applicable, targetable drugs. Though there are drugs which are available for FGFR1 fusion, but we don't have any specific drugs for FGFR1 amplification. I think all of us understand what a fusion is and what an amplification. So, in this particular patient, right now we cannot offer any of the targeted therapy. So this patient was actually started on conventional chemotherapy. Supposing if this particular patient down the line develops progression, then when we repeat the biopsy, perhaps at that particular point of time, I think I have not pasted the allelic burden of this. If I correctly remember, the FGFR1 amplification has around 25% value. That means we cannot just ignore it as a non-driver. Probably it is a driver, or probably that is one gene which is the cancer is addicted. But right now I don't have any targetable agents. Now, had this patient had harbored an IDH1 mutation, because IDH inhibitors are now available. Had this patient harbored an IDH1 mutation, then probably we could have considered an IDH1 inhibitor available right now commercially. So this is one particular report which I actually wanted to put a patient's lung cancer patient report, but somehow. But this is one way which we do it baseline for all the patients. Not so good. Let me come down to the reality of all this. So one important thing about targeted molecules, all these small molecules, are the off-target effects. And these when I say off-target effects, these off-target effects are seen across so many things. For example, the most common side effect with all those erlotinib, gefitinib is rash. And rash, seborrheic dermatitis, and significant amount of diarrhea, because these TFR receptors are expressed on the skin as well and on the gut. These patients, they have a very high incidence of diarrhea and rash, but manageable. There are drugs for managing those side effects, and long-term side effects with respect to the metabolic side effects. So these patients usually 3 years. Take the case of CML for 5 years. These patients are on the small molecule inhibitors. What metabolic consequences will they have? What is their effect on the patient's lipid profile or lipid status? What is their effect on the vascular status? Are they more prone for or vascular? What is the effect on long-term effect of GFR, that is creatinine clearance of? So now, since we have evolved for the past 10 years, now we are going. We are more and more seeing these long-term side effects of the small molecule inhibitors, because there's no drug without any side effects, obviously. So now we are understanding more and more about these long-term side effects. And coming to the costs of therapy, access to these drugs in Indian scenario is 10%. If you ask of me the Foundation One Medicines test, which we did, the cost of this test itself is around 2.7. Patient was very insistent on doing it. That is why we had to do it. But just imagine how many of our patients actually can afford this. It's not the therapy, it's the cost of this test, the NGS. So development of indigenous or institutional protocols. Because I really did not want to test all those. So development of our own institutional protocol, our own NGS panels, which we have succeeded in doing it for our patients with lung cancer, colorectal cancer, breast cancer, and so we have our own institutional protocols where we know that identifying this driver is very important because we have a targeted therapy against that, and it is cost-effective targeted therapy rather than just going ahead and doing the foundation test where they test for 500 genes. That cost-effective analysis, or rather, choosing what is right for our patients is also very, very important. Last but not the least is the clinical trial. Something is very important for us, and slowly have a clinical trial group across India where we have patients, because we actually for patients malignancy, our patients who are on long-term treatment, considering the option of enrolling them into clinical trials is very, very important. That is how our drug discoveries happen, because we need clinical patient population drugs or the therapy. So the way ahead, I'll just summarize identification of an ideal target with specific, effective, and safe. That is one of the very important crux of all this. Of course, development of ideal molecules, increasing access to both the diagnostic and the drug. I just back to that Foundation 1 report, and I told you, had the patient harbored an IDH1 patient, would have to offer him an IDH1 inhibitor. Now, the cost of that IDH1 per month is 6, and with the test, that is 3 lakhs for that test. That it was affordable, so we had to offer them everything. But I would leave it to you how many of our day-to-day patients actually think about that. So increasing access to the diagnostics and the drugs is very, very important. So here comes the role of state controlled, or rather state assisted health policy. So and of course on the translational research, developing our indigenous and like institutionalized testing. So we have taken the first step in our own diagnostic panel rather than just doing a gamut of sequencing. With this, I would thank you very much.
Great talks by both of the speakers. Purnachandra, drug will be identified and then taken forward over. Brilliantly showed the beauty of his talk was, I think, for the Indian, what we can do, and quick questions and
the panel which you mentioned, we are doing it now. When we do lung tissue sample, the bronchial biopsy, first guided biopsy, but the histopathologist, targeted characteristic, as you said, 30% also have got full genocide.
So we start off with incidents that have an there was in which we analyzed around 700 odd patients with cancer. All of them were subjected for instrumentation testing. We had around 35 to 40 patients who had. Now, so should we subject it for every patient? Probably. Because 10% of the patients still have one of those. Standard first line treatment still remains chemotherapy. But at the time of progression, we exhausted all the lines, then this will come.
Because now your technique where we can do a larger tissue, like IO-probe actually.
also. For example, the T790M mutation, usually that mutation is acquired over a period of time when the patient is exposed to, it's one of the mutations. But there are a certain set of patients where this T790 doesn't even. So people have tried to analyze why this happens, and they have found out that it is the germline transmission of T7. So nowadays, the patient agrees, you also send germline T790. But as you have asked, apart from smoking, what could be the carcinogenic factor? Probably it's unknown still.
So I have a question. In the NGS panel, the sample is or biopsy sample or blood sample?
Because at the baseline, we do the tumor tissue. At the time of progression, sometimes what happens is it's very difficult to get the patient might not agree. Then the liquid biopsy comes to rescue. But the caveat with that is we will not be able to confidently determine allelic burden of the mutation. And we will not be able to do the PDL1 because PDL1 is biomarker for immunotherapy. That is the caveat, but by and large, it is always a tissue.
So then the problem is small chunk of tissue. Right of
Yes,
it. So if there is tumor heterogeneity, you may not be picking up.
Yes, sir.
Yes,
sir. Again, to support your thought, there have been people who have actually biopsied 2 different metastases, and they have found certain drivers which are not there in it. For example, we have a liver lesion and a bone lesion. So the driver in the liver lesion and the driver in the bone lesion, there will be commonality, but there might be some subtle differences. So all of us are just in the as to how to handle this wealth of information.
You can. Thank you very much for